into cell communication and cell signaling today. And this lesson is pretty deep, so hang with me here through this. But we are going to, just for just a second, go back in what we talked about a little bit yesterday, because you got to get this in order to get the next piece. So we are going to be talking about these signaling pathways and the receptors that are responsible for these signaling pathways. But I just want to revisit the 3 stages of the signaling pathways are reception, transduction, and response. So reception is when the signaling molecule binds to that receptor protein, okay? The transduction are the series of changes that follow that, okay, which are going to include a shape change, okay? When these molecules bind, they bind and they change shape. They bind and they change shape. They bind and they change shape as they get activated. And then the last thing is going to be the response. So a cellular response. That can be lots of different things, okay? That can be turning on something. That can be turning off something. That can be opening a gate. That can be closing a gate. That can be making a protein. So signaling for a protein to be made, or helping or stopping or enhancing gene expression. So lots of different things could be the cellular response. Okay, so we're going to talk about a few different types of receptors that are in the cell membrane today. So the first one I want to talk about is the G protein coupled receptor. So it's sometimes abbreviated as GPCR, G protein coupled receptor. So this is a picture of our cell membrane, and this is the G protein coupled receptor in the cell membrane. Okay, so it's a protein, right? You see this structure here, which you should recognize as the alpha helix. That is in the secondary structure of a protein. Okay, and then it's also kind of like, coiling up here. You might say this is a part of the tertiary structure, okay, the way that it's interacting with those R groups. But at the top here, we have our binding site. So this is where a molecule, sometimes referred to as a ligand, okay, a signaling molecule, a ligand is kind of a generic name for it. You may see that or read that at some point, is going to bind here in that binding site. Okay, so this is a transmembrane protein because it spans the whole membrane. It's also an integral protein because it's integrated in the membrane. It fits both of those criteria. So this is the G protein coupled receptor in the cell membrane. Okay, it's going to bind things. This is the outside of the cell. This is the inside of the cell. So things are going to bind on the outside. Okay, and then they're going to be processed. The signal's going to be sent inside the cell. Okay, so here's our G protein coupled receptor in our cell membrane. Okay, and it's going to have a signaling molecule that binds to it. Okay, and so when that signaling molecule binds to it, it activates the G protein coupled receptor. Okay, generally when a molecule binds to something, that causes a shape change. Okay, bind equals shape change. We got to know that. Okay, and it also equals activation. So this is causing a shape change, and it's going to activate it. So this activated G protein coupled receptor is going to then bind to the previously inactive G protein. So you see here, this G protein is inactive. It's got GDP bound to it. Okay? So when it binds to it, it causes GDP to convert to GTP, and that's going to activate that protein. Okay, so now let's pause and talk about this GDP and GTP. Okay, so you've heard of ADP and ATP. That's adenosine triphosphate or adenosine diphosphate for ADP. Remember, ATP is our cellular energy. If we break down ATP and we break it down into ADP, we lose a phosphate, okay? And it releases energy. So when we add a phosphate to something in the process of cell communication and signal transduction pathways, I want you to know when you add a phosphate to something, it is generally going to activate the molecule, okay? So the G protein, just to come back up here to our notes, coupled receptor binds to an inactive G protein, which is what we saw here, causing GDP to convert to GTP, which activates that protein. So when that GDP gets converted, that is going to activate this G protein, okay? And so then that G protein can go on and affect something else. And so what it's going to affect is this inactive enzyme, okay? And so that activated G protein is going to bind to that enzyme. When it binds, we have a shape change. And you see that here. This inactivated protein kind of looked like a backwards C. Now it looks a little bit different, okay? It caused a shape change. And then that activated enzyme may go on and elicit a cellular response, okay? When the phosphate comes off, everything goes back to being inactivated again. When the signaling molecule is removed and the phosphates are dropped, it looks like it did up here in step 1 at the start, and everything is inactivated again, okay? When those molecules leave, when the it is dephosphorylated, okay, when that phosphate is removed, okay? We got that there? So the G protein coupled receptor in the cell membrane, the signaling molecules binds to it, that activates the G protein coupled receptor, which is going to bind to the G protein. The G protein gets phosphorylated, turning GDP into GTP, which is going to activate the G protein, okay? The G protein will then go on to activate this enzyme. And once that enzyme gets activated, it can go do something else and elicit a cellular response. So here we have our signal, okay? This step is a transduction step, okay? Actually, technically, this would be a transduction step 2, all right? The G protein, and then the response down here, okay? So signal, transduction, response. That's a cell signaling pathway. So that's the first type of receptor that I want you to know about. Let's look at the next one. This next one is a receptor tyrosine kinase, okay? And so kinases are enzymes because they end in ACE. Anything that ends in ACE is an enzyme, and their job is to transfer phosphates. Transfer that P, okay, like in ATP, ADP, GDP, and GTP. It transfers phosphates, and specifically receptor tyrosine kinases are going to transfer phosphates to a tyrosine, okay, to a tyrosine. So we have our signaling molecule here, which is our ligand, and it's going to bind here in the site, okay? And so these tyrosine kinases, right now they are inactivated, and there are 2 separate one of them. They are referred to as a monomer, okay? Once they come together in this structure over here, that's referred to as a dimer. But they can't do that until they both receive a signaling molecule. So once the signaling molecule binds, to both monomers, then those monomers will come together to form a dimer. Okay? Now, that dimer is inactive because it hasn't been phosphorylated yet. It hasn't had the phosphate added onto it. So adding a phosphate, remember, activates. And that process is called phosphorylation. If you remove a phosphate, it deactivates. And that process is called dephosphorylation. Okay? So, then this tyrosine dimer, in order to be activated, it's going to have some phosphate on it. So we actually need 6 ATP to do that job because there are 6 tyrosines that need to be phosphorylated. Okay? And so that's going to fully activate this tyrosine dimer. Okay? Which can then go on and bind some proteins that's going to elicit a cellular response. Now, one thing that is unique about the receptor tyrosine kinases, which is different from the G protein coupled receptors, is that you have not one but 2 cellular responses that can occur. Okay, so the receptor tyrosine kinases are important because they can activate multiple relay proteins and cause multiple cellular responses from the same receptors. Okay, G protein coupled receptors can't do that. If we look if we look back at our G protein coupled receptor, it just had one cellular response because there was one enzyme that got activated. But back over here, 2 different enzymes got activated because they can bind to those phosphates. So technically we can have more because there's plenty more phosphates left. And so this can cause multiple cellular responses. Now, receptor tyrosine kinases, this is outside of the AP exam, but I'll just tell you. Receptor tyrosine kinases are really important in breast cancer. So I don't know if any of you all know anybody that have had breast cancer, but typically breast cancerous cells have lots of receptor tyrosine kinases in them. Those irregular cells seem to have a lot of RTKs in them. And so treatment for breast cancer frequently focuses on signaling molecules that will bind to these RTKs and stop their activity or deactivate them. And so that's kind of a relevance here, just to give you a little bit of, why is this important kind of thing. Okay? So we've got G protein coupled receptors, receptor tyrosine kinases. The next one that I want you to know about are ion channel receptors. So ions are really important in muscle contraction, okay? And so when these ions move through this gate, here in this picture too, they will cause a cellular response. When the ions flood and move in the cell, they'll cause muscle contraction. That is our cellular response here, okay? Now, we don't want our muscles to be contracted all the time. Okay, that would be very painful. You'd have cramps or Charlie horse or whatever. Okay, and so this gate cannot stay open all the time. That would be bad. And so we have to have a signaling molecule that causes these gated ion channels to open and close. And so this is another example of a signal transduction pathway. So we have our signaling molecule that'll bind. When we bind, we have a change in shape, okay? And so that change in shape is what's going to open the gate, okay? When that signaling molecule binds, then the gate will open, allow those calcium ions to move inside, which is going to go on and cause a cellular response of muscle contraction, okay? when that ligand or that signaling molecule is removed, you'll see that the gate closes, okay? Because if you add that molecule or take it away, you're going to have a change in shape, okay? So as soon as those molecules are removed from any of these three, everything becomes deactivated and it goes back to the original way that it was, okay? And these are just another type of protein channel in the cell membrane. These are specific for an ion, and in this example that I'm discussing, it's a calcium ion. Okay, so that's an ion channel receptor. Last one I want to talk about is an intracellular receptor. So I believe I mentioned yesterday that these receptor proteins may be on the cell membrane, like we saw in the ion channel, like we saw in the receptor tyrosine kinase, and like we saw in the G protein coupled receptors. All of those were in the cell membrane. Okay, I'm drawing a line there in the phospholipid bilayer. But what we see here is the receptor is not on the membrane. The receptor is actually inside the cell. It could be in the cytoplasm, or it may even be in the nucleus. Okay? Now, the reason most of them are on the cell membrane is because many times the signaling molecules are big. Okay? And if you think back to what we learned in the last unit, big molecules have a hard time getting through the cell membrane, right? It is there to keep things in and out. And so those big molecules can't get in easily. And so if it has the receptor on the surface, then it doesn't have to go in. It just binds to the surface and causes the change that happens inside, that's relayed essentially inside, okay? But in some cases, such as in this case with our testosterone hormone, it's inside the cell. Okay, so we talked about hormones yesterday. That's a long distance signaling. That's an endocrine signal. Hormones travel through the blood. They take a little bit longer to act, but they also have a longer response. And so as this hormone diffuses through the cell membrane, then it will bind to a receptor. Okay. Now, not all cells are going to have a receptor for this testosterone hormone, but some of them will, okay? And these ones that have a receptor for the cell, for the testosterone hormone can go on and have a response. So in this one, the response is to go into the nucleus and impact the process of gene expression, okay? So this is where in the nucleus where the DNA is, which is used to make the RNA, which is used to make the proteins, okay? And so an example of this would be like sex characteristics. Okay, so when these genes get activated by the hormone testosterone, it turns on proteins that cause males to have chest hair and, have deep voices and produce things that females don't have. It causes gene editing or gene expression when that testosterone hormone binds. Okay, this receptor that can then go bind to a specific spot on the DNA and allow for gene expression of those things. Okay, if you have the hormone estrogen, it's not going to bind to this receptor because remember, estrogen is a different shaped hormone, and so it's going to have a different fit. The receptor and the hormone or the signal that binds are very specific to one another, and that's really important. And only cells that have a receptor that fit the signal can respond. Okay, that's also very important. So this is an intracellular receptor. It's inside the cell, potentially inside the nucleus, and it's going to, oftentimes it will regulate gene expression, will be the cellular response, which means it's going to control what type of proteins are made. Okay, so I think that hits all of that for today. You all are going to be working on your stop motion videos today and then tomorrow. So I think the worksheet that I gave you says to choose, it's the activity 11.1 worksheet that's in your binder. I think it tells you to do both of them. I only want you to do one stop motion video. So you're either going to choose the G protein coupled receptor or the receptor tyrosine kinase. And you're going to walk through with clay or paper models or however you choose to do it. And you can download a stop motion app. I downloaded one on my phone. I have an iPhone, and it was a free app, but the picture of it or the little icon is like turquoise, and it has a little movie reel kind of thing on it. But you'll move the clay, and you'll move, like you would if you were making a comic, a flip book, so to speak. And you'll take pictures of it, and you'll put those together in a really fast strip to make a stop motion video. And it will show the process of how this happens, how the signal transduction pathway occurs for receptor tyrosine kinases or G protein coupled receptors. So you have the rest of the day to work on that, or Friday as well. So those will be due on Monday, and then there's 5 questions on that worksheet for you to answer along with that. So if you have any questions on the stop motion, you can send me a message and let me know, and I'll try to help answer them as best I can. Y'all have a great day.